Today I am talking about the first topic of bioinformatics, aims and tasks of the bioinformatics. And I'm Satya Ishwari, Associate Professor, Biotechnology Department, NIT Raipur. So, as we seen, what is a bioinformatics? Bioinformatics is an interdisciplinary science that develops methods, algorithms, software tools to collect, store, organize, analyze, and interpret biological data. So the biological data here means DNA sequences, protein structure, and RNA sequences, protein structures, gene expression profiles, and generated by moderate laboratory technology like different DNA technologies. In short, the bioinformatics definition is that biological data is usable knowledge. How to use a biological data into usable knowledge by writing the software and building databases that make large scale biology possible. One human genome contains 3 billion DNA letters, so it is very much high information to understand my manually. So we need to something to understand and something to use the data, analyze the data, and process the data. And sequencing cost have been fallen faster than computing cost have computing risen. So sequencing cost, it is taking this much time and this much process, but computing will be computing is very faster because it is less costly and easier, little better than sequencing. Biology became a data science almost overnight because of this much data, so much of information, so much of 3 billion data letters are there, DNA letters are there in human genome, means it is how we can expect that. So what is a bioinformatics? In the NCBI 2001, it is established National Center for Biotechnological Information, the field of science in which biology, computer science, and information technology merge into a single discipline. Like biology is having so much of data, and computer science will be using all the databases and all the computational algorithms. We will use that algorithms and we will try to understand our data and analyze our data and conclude our data with existing computational technologies and information technology, whatever algorithms, what is the all the databases we will use. And there is Fred Tekaja. He is defined as the mathematical statistical computing methods that aim to solve biological problems using DNA and amino acid sequences and related information. Okay, so mathematical and statistical computational techniques can be used to understand our data, if we understand the data, so we can able to reduce our experimental efforts very fast. So how is bioinformatics came to be? Okay, how bioinformatics came to be? A short timeline of the milestone that shaped the world is 1953 DNA structure was invented by Watson and Crick. 1970 first algorithm came into the picture, Needleman Winch and other element algorithms. 1990 first human genome project was begun. It handled 3 billion base pairs of human DNA. And finally, 2003 genome is completed. The draft human genome is finished, creating flood data needing analysis. So if genome sequencing is completed, every data is stored in the database. We need to understand what the data contains, how the data could be analyzed. So today, big data biology, AI driven tool like AlphaFold, different cloud genomics, different techniques came into the picture. What are the origins and pioneers of our bioinformatics? Okay, what are the origins? What is the how bioinformatics came to be picture? Origins and pioneers of bioinformatics. Margaret Dayhoff was told in 1960 the first protein sequence Atlas and PAM scoring matrix, widely regarded as the founder of bioinformatics. And Genome GenBank came into the picture in 1974. It is the national laboratory begins collecting DNA sequences, creating the field first nucleotide database. Like how evolution of sequences of DNAs and proteins has been come into the picture. It became and alignment of algorithms started in 1970 and 1981. Blast and FASTA are the sequence alignment. It started in 1988 and 1990. More milestone along the way. This is the whole history of that. 1965, Dayhoff's Atlas was started. Margaret Dayhoff. He published first protein sequence by using the, and later it was PAM matrix was established. And in 1988, GenBank is a genomic database, and FASTA is the sequence alignment algorithm. NCBI is founded. Pearson Lipman published FASTA for the fast sequence similarity search. Similarity search algorithm first was published in 1988. FASTA is a similarity search algorithm. It's first published. To understand that, to compare what is a similarity search, to compare two sequences, when they compared and aligned together, we will understand what is the basic difference between how the difference is coming, where the mutation is coming, where the problem is coming, how the both sequences are different, why they both sequences have different function. These things we will understand. That the first sequence alignment algorithm FASTA was developed in 1988, and in 2001 draft genome published. The first draft of the genome, about 3000 mbp, is published. So this much data size is published. Genome means entire organism, entire one particular organ. Gene sequence, if you combine together and keep it, is called a genome. And then in 1981, local alignment is started. Smith-Waterman algorithm and alignment algorithms, this like FASTA is established by using comparing multiple sequences. Like that only dynamic programming algorithm came, comparing of two or more than two sequences by using similarity search. There is a local alignment and global alignment. So if we compare the short sequences, then it is called a local alignment. That local alignment algorithm, it is followed by one algorithm. That algorithm is called Smith-Waterman algorithm. It is established in 1981, and in 1997 microbial genomes were started. The genomes of E. coli and Baker's yeast are fully sequenced, enabled whole genome comparison. Microbial genome was started in 1997. Aims of the bioinformatics. First, whatever we talk about till now, so we are seeing about some sequence comparison. We are also seeing some, about genome, compare genomes established, microbial genome is established, human genome is established. So what to understand that data? Organize biological data, build a structural, searchable databases that store the ever-growing volume of genomic, proteomic and clinical data. It's not that you will keep the data, you will store the data without any meaning. It's not like that. We have to structurally store the data. Somewhere should be genes, somewhere should be genome, somewhere should be properly it is a proteomic data. It's a all proper data will be stored over there. Genomic, proteomic. It's like even we take an Excel sheet and we try to import some data, like something about physical characteristics and all. So properly we should organize. Otherwise next time we can't even take the file. So here also organization is very important. Organization in terms of genomic, proteomic and structural data. Develop analytical tools. we have to understand different algorithms will be created, different softwares will be developed to for sequence alignment, structure prediction and statistical model. Again, apart from sequence alignment, there is something called structure alignment. Structure prediction is also very important parameter. Structure, protein structures are predicted. Protein structures will be used for the function, because most of the proteins in our body, everything is built up of proteins. The proteins are structures. That structures only will be acting, showing some functional properties. Then like amylase, different enzymes are there in our body. So structural develop analytical tools to understand that data which is developed, which is reposited in the databases. That is the first aim of the bioinformatics. And the third aim of the bioinformatics, extract meaning. What we are trying to extract out of the data. Turn a rare sequence into structural output into biologically meaningful testable insight. Like what is, if you are developing an algorithm for sequence alignment, if you are comparing two sequences, what is your output of that? Maybe the first sequence belongs to diseased, another sequence belongs to healthy. So these two sequences, when we are comparing, we will understand that because the particular somewhere, maybe somewhere ORF start code and end code and somewhere the problem is there. That is why the sequence is becoming diseased. So we will understand. What is extract meaning? What enable discovery? Support research in genomics, medicine, agriculture and evolutionary biology. Advances in human health. Translated data driven findings into diagnostic therapeutics and personalized treatment plans. So finally we can come up with the personalized treatment. If you understand a particular diseased individual, healthy individual, we can come with the personalized treatment. Scope and branches of bioinformatics. Different scope is computational bioinformatics. In the computational bioinformatics, we try to develop write different algorithms, softwares to build curates public databases that score genomic and proteomic data. Applications of databases include sequence, structure, functional analysis, from mutation detection to drug targets. How we can target genomics and proteomics. Study the genome, proteome, transcriptome, three interactomic layers behind every cell's fate, because our whole central dogma is talking about DNA to DNA, DNA to RNA, RNA to protein. So whole entire concept, if we understand, entire gene we understand, we can entire proteome we understand, we can able to predict what is the problem, where it is coming, how to improve the productivity of one crop if you understand particular genome. Animal and plant bioinformatics that include agricultural, horticultural, medical and forest plant bioinformatics. And these are the different scopes of the bioinformatics. More specialized branches are there. Functionalized genomics, chemi-informatics. Functionalized genomics means functional relevance to genome scale data, studying genomes, their protein products and the biological roles these proteins play. Chemi-informatics, like organized chemical and natural products data, support structure-based drug design. Like chemi-information, chemicals are drugs like paracetamol, different structure it is having. Okay, so low molecular weight bioactive compounds. Based on function, if you understand whole genome, it is called a functional genomics. If you take a chemical structures of the chemical structures as an information and you try to conclude out of it, it is called a chemi-informatics. And it is a metabolomics, profiles and small molecules metabolites in cell tissues to connect genotype, like metabolic, like whenever we glucose to glucose 6-phosphate, glucose 6-phosphate to finally glycerol. So how glucose is as a metabolite, how it is converting into glucose 6-phosphate, why in the diseased individual it is not able to convert, why it is in healthy individual it is maintaining proper, which gene is responsible. That particular concept in terms of metabolic pathway, if you try to predict in genomics and proteomics, it's called a metabolomics. And it is another one is called structural genomics. Applies genome scale structure prediction comparison to characterize entire protein families rather than a single molecule. Structural genomics, entire protein families, whereas we talk about chemi-informatics, only single protein Okay. Core tasks of bioinformatics is first is sequence analysis. As I said, sequence analysis is nothing but comparison of sequences. Like when you talk about any gene sequence, we have ATGC, ATGC, ATGC. So we take a one sequence and we'll compare with the entire databases of the sequences. It is called a multiple sequence alignment, private sequence alignment. Sequence analysis will give us the where one sequence is entirely different with the other sequence. Then we make and find out mutations, other variations, so that we can have some conclusion out of it. Structural analysis, like protein structures are predicted. Protein structure, why we have to predict? Because prediction of a protein structure is an important parameter, because we have three different type of structure: primary structure, secondary structure, and tertiary structure. Primary structure is simple sequence, secondary structure is having some different helices and all. Third, tertiary structure will have a functional property, it can exhibit a function. So if any protein is doesn't have a 3D structure, it may not be able to exhibit any function. So tertiary structure, when it is there, like even in our body some enzymes are there, so many proteins are there, they will exhibit a tertiary structure and they will be having some function. That means they are able to bind with the some of the target in our body, so that they will have the capability of exhibiting a function. So that is where structural analysis is very important, to get the novel drug, to get the novel molecules, to understand, to disease prediction, cancer, this type of diseases, hormonal diseases, everywhere we can use the structural analysis. Data management, curating genomic, proteomic databases like genomic, GenBank, proteome. So data management is also important. If you are repositing your data randomly, irrespective of any proper, any, like I said in the first thing of scope of the bioinformatics is analyze the data and curate the data and keep it in, store the data in proper places. That is very important. Evolutionary analysis, after doing a sequence alignment, after doing the structure prediction, you have to compare with any other sequences existed in the universe similar to this sequence, similar to my organism, similar to my unknown gene, similar to my unknown organism. That evolutionary algorithm will, evolutionary analysis will help us. Sequence analysis and alignment talks about pairwise alignment and genome annotation, pairwise and multiple sequence alignment. Variation, mutation detection, in this we will try to understand, like these two sequences are there, how both are different, where is the mutation coming. Okay, so this way we will try to understand. Structural bioinformatics talk about structure prediction, molecular docking, comparative modeling, structure visualization. We can compare two structures and we can see how the better this because based on the structure, this can be a better drug than other drugs. Genome and database management. So while repositing our data, we should reposit raw data. We may deposit secondary data. Secondary data means pre-processed data. So that genome and database management is also very important. Like GenBank is having, it is a nucleotide database. Protein databank is having protein structures. It is having protein databases. Evolutionary comparison, we will try to compare species A, species B. What is the relation? If we isolate any gene from the soil, what is the relation with our unknown gene with the existing thing? Without doing any experiments, we are able to establish. Applications in real world, drug discovery, personalized medicine, agricultural, public health, forensics, evolutionary studies. Broader applications of bioinformatics include gene therapy, preventive medicine, waste cleanup, climate change studies, alternative energy, veterinary science. Challenges and future directions are data volume. Volume is increasing day by day. Data is increasing day by day. How to handle the data, how to store the data, how we can use the previous data, existed data to know. Privacy and ethics, genomic data, deeply personal, sharing it for research and must protect individual privacy. Standard privacy is important if we are sharing our data, so it should not be used for the wrong purposes. That is why privacy is the matter. Standardization is important. Formulas and disciplines pipelines vary across the labs, making results harder to compare and reproduce. This is the whole concept about bioinformatics turns into food of biological data, genomes, proteomes, beyond, into usable knowledge. By combining biology, computing, statistics, it organizes information, builds analytical tools, and enables discoveries that drive medicine, agriculture, and our understanding of life itself. Thank you.